Immunology. Today we'll be talking about this process of T cell activation, and I find that the easiest way to explain this is by giving a clinical example for you to understand. So we have a 28-year-old male who presented with rash in his hands and came to you, and the first thing you're going to think about is, all right, so that person is having a rash in his hands, and I just want to understand how that rash occurred or why that patient is developing that rash. So, in simple terms, the first thing that will happen is there's a specific antigen. That antigen might be infection, or it might be an autoimmune response. His immune system is mistakenly attacking his skin cells due to an abnormal processing or an autoimmune disease that led to that. Um, but the first step will always be that there is a specific antigen, and that antigen was being detected by the antigen presenting cells in the body, like for example the dendritic cells, and then the macrophage, and the antigen presenting cell traveled to the closest lymph node where the T cells are, and they presented that antigen to the T cells in order for them to get activated and come to help with the inflammation process. So I put everything here to you in one picture. So everything starts here actually, where the antigen presenting cell came to the closest lymph node where the T cells are, and they are carrying the antigen, the information of that antigen to the T cells. At that point, there is a very critical reaction that should happen and if that reaction doesn't happen, all the other steps will never occur. And that reaction is the reaction between the T cell, the CD4 T cells, and the antigen presenting cells. They should a certain connection should happen between a protein called the B7 protein on the antigen presenting cell with the CD28 receptor on the CD4 T cell. And the MHC class 2 receptor should combine with the TC receptor and the CD4 molecules on the surface of the CD4 T cell. If that reaction or that connection does not occur, all the subsequent chemical reactions will never happen. So the first step on T cell activation is the combination between the B7 protein and the MHC class 2 with the CD28 receptor on the T cell and the TCL and the T cell receptor and CD4 molecule on the CD4 T cell. If that reaction happened successfully, the T cell will become fully activated. And the process of the T cell activation, CD4 T cell activation, will eventually lead to three main things. Number one, B cell will be activated to produce more antibodies, which will explain, of course, why there's this rash in the patient's skin, because IgE, for example, is one of the things that are connected to allergy, which is produced mainly by the B cells and by IL-4, just like I'm going to explain to you in a minute. And it's going to stimulate the other form of T cells, which is the CD8 T cells, and then we're going to activate the macrophages. So I know one of you will say, all right, but how come the macrophages are not activated right now? But they are, but we have a really important thing that you have to understand is that sometimes although the macrophages are being activated, but the T cells also will have the capacity to reactivate the macrophage, and the macrophage will have the capacity and the ability to activate the T cells whenever they need to. So eventually this will happen over here. After all these reactions, eventually this will happen over here. Production and activation of B cells will occur, stimulation of the CD8 T cells molecules will occur, and the activation of the macrophage will occur. There is two different types of CD4 T cells. The first type is called the Th1 CD4 T cells, and the second one is called Th2 CD4 T cells. And they affect each other in a lot of ways, as I'm going to explain to you in a minute. But you have to understand very, very important concept that CD4 T cells, they're not only one type of cells. No, there is two types of CD4 T cells. The first one is Th1 CD4 T cells, and the second one is the Th2 CD4 T cells. We're going to start talking about the Th1 CD4 T cells. They the Th1 CD4 T cells will always try to stimulate this pathway, stimulating itself and inhibiting the Th2 CD4 T cells. And the opposite is true. The Th2 CD4 T cells will always try to stimulate itself and inhibit this Th1 CD4 T cells. So they always try to inhibit each other's, and that's a very important thing because as far as we need inflammation and we need our immune system to fight an infection, for example, or a pathogen, for example, but we don't want the immune system to get too crazy and initiate a very strong immune system that might even damage the human cells, as I'm going to explain to you in a second. So we are talking about the Th1 CD4 T cells. They produce mainly four different types of interleukins per se, or like not an interleukins per se, but some sort of inflammatory mediators. Number one is the IFN gamma. The interferon gamma is the one that's going to activate the macrophages more. And the thing is, why I started talking about the interferon gamma first, because interferon gamma is going to try to inhibit the Th2 CD4 T cell for the same reason that I just told you about. So it's really important for you to understand that Th1 CD4 T cells will produce IFN gamma, which is eventually going to try to inhibit the Th2 CD4 T cell pathway, and actually it's going to also try to activate more macrophages. The second thing that's going to be produced is the interleukin 12. And this is a very, very important interleukin for you to remember because it is not only going to activate even more macrophages to come to the area, but it is going to stimulate the Th1 CD4 T cells to be produced more and more. So the interleukin 12 will be produced by the Th1 CD4 T cells, and when it is produced, it's going to produce more macrophage and send another feedback response to produce even more and activate even more Th1 CD4 T cells. The third thing is we're going to activate the CD8 T cells. And the CD8 T cells are very well known that they are responsible to produce IgG, one of those antibodies. And the thing is that you need to understand is that it's critical to produce the IgG in a lot of diseases because the IgG has the capacity to help with a process that we call a cell-mediated immune response. Cell-mediated immune response means that sometimes we need to kill the human cells because pathogens lives inside it. Meaning, sometimes you have diseases like tuberculosis, for example, or listeria. Those pathogens, they live inside our cells. Tuberculosis lives inside our macrophage, for example. So at that point, we need to kill those macrophages so we can get rid of the tuberculosis. And the only way for us to do that is activating the CD8 T cell molecules because they are able to initiate a cell-mediated immune response, which means they can destroy the human cells itself. The fourth reaction will be the interleukin 2. The third cytokine produces interleukin 2. Actually, interleukin 2 is a very important molecule because not only because it's a T cell growth factor, which means it can even activate more CD4 T cells and more CD8 T cells, but also is able to activate the B cells and natural killer cells, which you know that natural killer cells has the capacity to kill the tumor cells in our bodies every day. So that's why we now have a medicine in the market that's called the aldesleukin, which is an interleukin-2 chemotherapy agent, which we use to treat renal cell carcinoma whenever there's cancer in the kidney. One of the medications that you might see the patients are taking, or the chemotherapy agents that the patients are taking, is aldesleukin because it has interleukin-2, which can produce more natural killer cells, which has the ability to kill tumor cells. So this is the first pathway, the Th1-CD4 T cell pathway. The second type of CD4 T cells are the Th2 CD4 T cells. And they also produce another cytokines, interleukin 4, interleukin 5, and interleukin 10, and they themselves produce help activating B cells to produce more antibodies. The thing is that you have to understand that we always need interleukin 4 because it has two main jobs. Number one, it increases the expression of IgE, which is well known to kill any parasite, but also it has the capacity to produce and activate the Th2 CD4 T cells and inhibit the Th1 T cells, again, because we don't want too much of an immune response at any time, because we don't want our body to our immune system to attack its own cells. So at some point, we don't want the immune system to kind of overreact to a pathogen, because that might lead to a damage more than good. So the interleukin 4 will stimulate the production of IgE, which is going to kill any parasite in the area. It's also going to activate the more Th2 CD4 T cells and inhibit the Th1 pathway, or the Th1 CD4 T cells. And then you have the interleukin 5, which is well known to produce IgA, which is a very important molecule and or antibody to kill all the GI bacterial infection, gastrointestinal bacterial infection. And also, it increases the expression of eosinophils, which is very, very connected to allergy. That's why you see this kind of allergic reaction in the skin, because of interleukin 5. Now, we understand that at some point we need that patient, after this reaction happened to the pathogen, we need the immune system to try to relax and stop the immune response, because again, we don't want too much of an immune response at any point, at any time in life, because too much of an immune response can lead to more damage than good. So that's why we have interleukin 10. Interleukin 10 has the capacity to shut down the whole process, and that's why it's very important to stop the inflammation by the production of interleukin 10. And that's how you see this allergic reaction or the immune response getting CD4 T cells activated.